Yeah, that's the end. Like, oh, that's it. Yeah, yep, that's it. Okay, that's it. Okay, so these were our four, and so this is what when we put the plasmid in. So you have some transformed bacteria and some that are not. You sucked up 100 microliters and you put it on the LB/amp. Simon, turn around. LB/amp petri dish, and then you spread it around with the loop. You put it in the incubator where it's nice and warm, and you should have had growth. So everybody should have seen growth on this plate. And so my question to you is, what bacteria grew on this plate? Which type, the transformed or the non-transformed? The transformed. Why? Because any bacteria that did not take in the plasmid would not then have the ampicillin resistant gene, which means then they wouldn't be able to make the protein that breaks down ampicillin and makes it be able to survive. So when you put these on there, you put both kinds of bacteria on this plate, but all the bacteria that weren't transformed all died due to the ampicillin. Only the ones that have the plasmid was able to divide and make colonies, and so the colonies that you see on there are colonies that have that plasmid. So there's what you can't see is a bunch of dead bacteria on the top, okay? So then, the second one was the positive P glow with LB, AMP, and ERA. So therefore, the only difference between this one is that it has arabinose in addition to LB, which is the food, and ampicillin, which is the antibiotic. So in this one, you should have also seen growth. And so for the same reason, for this one, you took 100 microliters and put it in here. You put transformed and non-transformed bacteria, but you should have only seen on your growth, you see only the transformed bacteria. And the only difference between these two is the arabinose. What does the arabinose allow to happen? Yeah, the fluorescence. And so what is fluorescing? So it causes the fluorescence. What is actually, yes, but what's actually causing that to happen? The presence of?
Dropping of energy.
Where's the energy change and what molecule is that happening? Is the DNA glowing? Is the gene glowing? Think about this. If the gene was glowing, what would this one be doing? Glowing as well, because this bacteria here also has the green fluorescent protein gene. So it's not the gene. So this one is doing what with the gene? It's going through protein synthesis, and it's a green fluorescent protein gene, which codes for making a green fluorescent protein, all right? That fluoresces green. So it's actually the protein that's actually causing this to happen. That's why this one isn't glowing. That's also why when I walked around yesterday with that little vial that just had the plasmid in it and shined the light on it, that shouldn't have glowed. There's no protein in there. It was just the DNA. So it's interesting because some people, I think, were trying to psych themselves out because they thought that they were supposed to see green. So throughout the day, I had people going, "Oh, I think I see a little bit of green in there." There's no green in there. But that's what you were thinking, what you should have seen. The DNA itself does not glow. It's the protein. And that's why this one doesn't glow and this one does. These two bacteria are genetically identical, these two. They have the same DNA in them and so on, so they're identical. This one over here, could we make these glow?
Yes.
What would we have to do to make them glow?
Add
arabinose, and that's what I talked about on Monday. Remember arabinose, the arabinose operon, and so it turns on that gene. All right, absolutely. So that's that. Then these two, we put the negative P glow, and here's your vial. So the negative P glow vial. The only difference between this one and this one is that this one just had the bacteria and it doesn't have the recombinant plasmid. So that you never came up and you shouldn't have come up and put a loop full of the plasmid into it. So it just has the bacteria. So now we plate it on LB in amp. Should we see growth on this one? You took 100 microliters of this, put it onto a plate that has LB, which is food, ampicillin, which is an antibiotic. Do these bacteria in this negative Pliego tube, do they have the ampicillin-resistant gene? No, and you never gave them even a fighting chance because you never put the recombinant plasmid in there, so there's no way it could have that. So therefore, you should see absolutely nothing on this plate, all right? And that's the reason why. Then this plate here, this plate just has food, so for the bacteria. So now you put 100 microliters on here. Is there anything that's going to kill these bacteria? No. This one right here, you should have the most growth. And the growth should be so much that you can't even count the colonies. So when you did this yesterday, and you put the 100 microliters on here, what happened is when you spread it around with the loop, there was so much bacteria. So there's a whole bunch of bacteria you couldn't see, but that you were spreading it around. You put it in the incubator overnight, that bacteria starts to divide, and these colonies, there's so many bacteria that when they divide, they're making these colonies. All the colonies kind of all, merge together. And so what you should see on the plate looks like one big smear of bacteria. And that's so many bacteria that the colonies all are together, okay? And so that's what we should see there. Okay, you guys see that? Okay. So that's what you should have seen. So let's take a minute and talk about the lab right up here. This sheet I gave you on Friday with the other lab stuff. It says AP Biology Formal Lab Report format. This is the same format we've used for other lab reports, so I'm not going to go through every section because we've already done that, but I'm just going to illustrate or highlight some of the things that are different about this. So you have your title, you have your background information. With your background information in bold there, I put specific questions that pertain to this lab that you should include in your background information. These are things that somebody would need to know to be able to understand what's going on in your lab. So make sure you answer those questions within that background information. Then the purpose and the variables, that's the same, there's nothing new to say there. When you look at the opposite side, the hypothesis, notice the last sentence underneath there, underneath that section for hypothesis in bold, it says for this lab, you may attach your pre-lab prediction chart. I'm going to let you, because you already made a hypothesis, I'm going to let you use that chart that you did together in class on Monday for that. But I still, in your typed out lab report, want a section that says hypothesis, and then next to it just say see attached handout, okay? Yes. Hypothesis. All right, so see attached handout there. Then we have our materials, that's self-explanatory. Then, hopefully this will make you happy for procedure. Look what I'm going to let you do. For this lab, you can attach the one-page handout. All right, so that one page that you used yesterday, front and back, I'm not going to make you type out the procedure. So you're going to write for procedure, same thing, see attached handout, and put it on the back. You're welcome. All right, so then data and observations. When we get to that section, you are not doing a graph for this lab. What you are going to do is kind of like a data table, kind of similar. You can do something similar to the sheet that you just wrote on that I passed out or handed out today. Remember, this is where you can embed you're going to have information for each plate, and you can embed the pictures if you took pictures. And then I want you to write down information about what you saw. So, all right, so information about the colonies and so on. So very similar to that sheet, but you are typing it out and putting it in. So you're not attaching that sheet that you filled out today. All right, and then conclude. The rest of it's pretty self-explanatory. All right, so that is our lab report. Consideration 2, you were supposed to do in the downtime yesterday with your starter plates.
Okay.
All right, so if you can remember what the bacteria looks like and stuff like that. Okay. Okay, so that's that. And notice I put lab report due on Tuesday. So so that way you can focus on studying and then have the weekend to work on the lab report. Monday, if you come up with any questions that you want answered to help you with the lab report, and then it'll be due on Tuesday. So I'm going to give you plenty of time to do it. Okay? All right. Okay. So let's go on with our notes. So get those out, please. Now move on with chapter 20. One other thing, remember last week I went over this with you, I pulled everything together and told you last week exactly what was going to be on the test, what sections and things like that. So first hour today, I had a student ask me, she asked me if I could do review videos like last time for that. So I said yes. So during my conference hour today, I wasn't planning on doing it, so I had a little bit of time. So what I decided to do, I didn't think I had enough time to do all of it in one sitting. So what I did during my conference hour is I made little review videos. So I did 18.4 and did a little, that's operons. So you can watch that if you want to review about operons. And I did one about 19 as well, and so that's what I got done during my conference hour. So my plan is to do the other ones as well. And so they're going to be labeled. I already have the 18 and the 19 one uploaded onto YouTube. So so if you want to look at those, you can. But if not, that's okay too. And the sections, that way, if you want to just look at certain topics, you can too as well. All right, so without watching the whole video. Okay, so that's that. If you would like that. So hopefully tonight, I'm trying to do it in the time I have. So hopefully I can get the rest of it done here tonight as well. Okay. All right. So I just want to point that out. So this is where we left off. And this is the last picture here that we have. And so I'm going to have you don't turn the page yet, turn back to this picture here. All right. By this picture, either underneath it, I'm going to have you add a few bullets, these three here. And so either underneath it, some of you may have, depending on where you wrote the notes in the picture, you might have some room to the right of it. If not, you can go underneath it. let me talk about this. So when we put a eukaryotic gene into a prokaryotic cell, which is really what we did in this lab, that we put a gene from a jellyfish, which is eukaryotic cells, into the bacteria. The thing about bacteria is that they don't have introns like we do. So when we have introns in our DNA, our cells know what to do. They know how to cut out the introns and make mRNA that without introns. So this is what normally happens in our cells. So normally, this is a quick little review. Normally, we take the DNA, which has a gene on it, and that gene, remember, is part intron, part exon. Okay, so that's what we have. Then remember, we make a pre-mRNA strand from that during transcription, where we start at the beginning of the gene and go here. So this is your mRNA. It also has the introns and the exons. And then we go through that RNA processing where we cut out the introns and splice together the exons to have a shorter molecule. So this is your finished mRNA. And so normally in our cells, that's what's used for making proteins, and we cut out those introns. The thing is that if we want to take this gene and put it in a prokaryotic cell, if we cut the gene out like this and put it into a plasmid, so here's your plasmid here, we cut open the plasmid with the restriction enzyme and try to put this gene in and then put it into a bacteria, the bacteria is going to use the introns and the exons because it doesn't know to cut out introns because it itself does not have introns. So therefore, we as people had to figure out if we want bacteria to make a human protein or a jellyfish protein, we have to have DNA with no introns. Well, that doesn't exist in us, but what does exist in us with no introns? The mRNA that we make. The mRNA does not have introns. So what we have done is isolated the mRNA instead of the DNA. So we take the mRNA, and so this is a piece of mRNA. Here's exon, mRNA with no introns. Then in the lab, what we did is from that mRNA, made a piece of DNA from that mRNA that's made out of just exons. So here's DNA with no introns. Now how in the world do we do that? So what is it in nature that we know of? There's an enzyme that can convert RNA to DNA. Do you guys remember? There's something in nature that contains an enzyme that can take RNA to DNA. Viruses and viruses. HIV virus. Do you have a guess, Austin? No. I heard you whisper or something to Justin.
Oh.
You don't know?
Yeah, what's the first thing?
The first letter is an R.
Reverse transcriptase.
Reverse transcriptase is the name of this enzyme. So this enzyme, reverse transcriptase, is the reverse of transcription. Transcription goes from DNA to RNA. This is found in retroviruses, such as HIV. So this is an example of us taking something in nature and utilizing it. So we said, okay, this is what we have with no introns. How do we get DNA with no introns? What do we have available to us that can go from mRNA to DNA? Oh, reverse transcriptase. We isolate that from the virus, unleash it onto our mRNA, and boom, we have DNA with no introns. That DNA then can be inserted into the plasmid. So that DNA here can be inserted into the plasmid. So like this is the jellyfish gene that we just put in here, that had no introns. And so the company that we ordered it from, or I ordered it from, already did this part. They cut out the introns and so on and so forth. So they went through this process. Okay, and so that's that, and that can go into your bacteria, and then your bacteria can make the protein. And so that's what goes on. Okay? Alrighty. So the next over here. into a eukaryotic cell. So far we've just talked about bacteria, putting something in the bacteria. So a eukaryotic cell, yeah, oh, sorry. Yeah, I gotta go back. There we go. Thank you. So one method of introducing recombinant DNA into eukaryotic cells is electroporation, applying a brief electrical pulse to create temporary holes in plasma membranes. In addition to that, scientists can inject DNA into cells using small microscopic needles. So they use a high-powered microscope, look at the cell, and actually inject the DNA into the cell that they want to go inside. Once inside the cell, the DNA is incorporated into the cell's DNA by natural genetic recombination. That's another way to say crossing over. All right, so crossing over, natural DNA recombination. So bacteria cells naturally take up foreign DNA. Our cells do not. So we have to help DNA to get into our cells if we want to put DNA into our cells. And so this electroporation, if this is your cell, the electroporation just momentarily creates these little pores in the membrane. And so if you have foreign DNA on the outside, the DNA can actually go into the cells that way. If without doing that, if you put DNA outside the cell like this, the DNA will stay outside the cell. So that helps with the uptake of DNA. Or, like I said, we can physically inject it in there. And so then it does natural recombination. So let me give you an example here where, let's say this is your pair of chromosomes. And let's say somebody is r, where they have let's say that stands for a genetic disease, a recessive genetic disease. So what they've tried to do is to put into the cell a piece of DNA. So here's a piece of DNA, but instead of a little r in that same spot, it has a big R. And so what when it says they combine by genetic recombination, the thought is that if these two chromosomes, when they replicate and go through cell division, when crossing over occurs, that we can replace the big R with the little r, and therefore the person could have the big R gene instead of the little r gene, and therefore produce a protein that maybe they're missing, and therefore kind of a way to cure a genetic disease. We'll talk a little bit more about that at the very end of this, where I just give some examples of what we're doing with this, and that's called human gene therapy, where we're trying to, because really a genetic disease, the only way you can cure it is to change the genes, because it's caused by genes. So eukaryotic cells are a little bit different here. All right, another DNA technology thing that we use is something called PCR. It's polymerase chain reaction, they call it. Can produce many copies of a specific target segment of DNA. So there's a machine that does this. It's a 3-step cycle where it heats the DNA, cools it, and replicates it. And so what happens is you get this chain reaction that produces a ton of DNA. So this could be where if you want to make many copies of a gene, if you're studying a genetic disorder and you want many copies of it to look at, study the order of the bases and so on. Or let's say a small segment, a small portion of DNA is found at a crime scene, and let's say you have different scientists, like let's say the prosecutor's side and the defendant's side want to have their own scientists analyze the DNA, but yet you have a small sample. You can replicate it very fast using this process, so you can get lots of copies of that DNA. This is called PCR, and this is what actually happens in the machine. So what we have here, if this is your DNA, what we want to do is replicate it. So we're starting out, this is the DNA we want to sequence or to replicate, and then we end up from 1 to 2. So that's the overall big picture. So what happens in this machine is DNA replication. So what do we need for DNA replication? So you have to have the two sides of the DNA separate. That's the heating part. So that's the denaturation. So the hydrogen bonds break between the two sides. Then what do you need for DNA replication to happen? What enzyme adds bases? DNA polymerase adds bases. Can DNA polymerase add to the end of a molecule? No, so what else do you need? You need a primer. So things that you need. So what they do is, knowing about DNA replication, they put in this machine things that are needed for DNA to replicate. So what is needed for DNA to replicate? A primer. So we have an RNA primer. We need DNA polymerase. Remember, it adds the nucleotides. We need the nucleotides themselves, so we need to add 4 types of nucleotides. What makes each nucleotide different? The type of base that's added, so A, T, C, or G, and so on. So basically, we're putting in this machine everything that's needed for DNA replication, and look at what happens here. So the 2 sides separate due to the heating. Then it cools, and the primers then, here are the primers added on to the DNA. So remember this, so this is saying 5 to 3. So the primer here is, this will be the 5' end of the new strand, and the primer is going to move in this direction and grow in the 5 to 3' direction. This one here, 3' of the original, 5'. So the primer is added here, and the strand will grow in the 5 to 3' direction, and you end up with 2 strands at the end. And so then, what these 3 things, notice here it says cycle 1. So then here, this whole thing with these 2 strands, these 3 steps will occur again, and then we'll have 4 strands, and then 8 strands, and then 16, and so on and so forth. And so what you do in this machine is that you get many copies of a segment of DNA very quickly as a result of that, and that allows you to have lots of copies to study and so on. All right, and so that was a really big deal to be able to do that, so that we can learn more about segments of DNA. All right, and then the last new thing with the process of DNA technology is this restriction fragment analysis. Detects DNA differences that affect restriction sites. So I'm going to explain what this means. So restriction fragment analysis detects differences in the nucleotide sequence of DNA molecules. What makes them different is the order of the A's, T's, C's, and G's. So such analysis can rapidly provide comparative information about DNA sequences. So what does that mean? So everybody in here, we're all human, we have some DNA in common. So for instance, we all make hemoglobin, which is a protein that gets put into our red blood cells to transport oxygen, unless we have a genetic disorder that changes that. But so we all have that kind of gene in common, and so have that sequence of bases. But then we have other bases in the order of the A's, T's, C's, and G's that are different, and that's what accounts for differences between us. And so, so what we want to do is, what happens is that if we cut our DNA, so if I took a DNA from any 3 people in this room and cut them up with restriction enzymes, what do restriction enzymes do? Do you remember what they look for? They create sticky ends, but before they cut, remember they're looking for a certain order of bases. So, like for instance, CC, GG, and notice it goes the same back here. All right, and then let's say it cuts like this and creates. So that enzyme looks for a particular order of bases and cuts. And so, therefore, if I took DNA from any 3 of you, use the same restriction enzymes to cut up your DNA, would we come up with the exact same number of pieces, do you think? No, and that's because, is all of our DNA, the sequence, exactly the same? No, so some of us may have more cutting sites than others, and so therefore we end up with different sized fragments. So, so how we analyze that is using this process called gel electrophoresis. All right, thanks. That's So this gel electrophoresis uses gel, hence the name, and electricity. Okay, it uses the gel as a molecular sieve to separate nucleic acids or proteins by size. What's a sieve? Do you know what that is?
Yeah, something you can separate 2 things in a mixture by the size.
You can separate things in a mixture by the size. So, like we could use a spaghetti strainer as a sieve and put sand and rocks and stuff, and it'll let the sand go, but the rocks won't go through, and so on. So that's a sieve. And so So this gel separates DNA based upon size. And so that's what this picture here is showing you. So let's look at this. This is the gel. The gel is a thicker consistency even than the agar. The agar that we use is kind of gel-like. This is a thicker consistency. And we have wells on the top here that where you can put solutions in. So let me talk a little bit about this. This gel, first of all, has pores in it. So microscopic pores. You can't see it when you look at the gel, but it has little small holes that things can move through. And what is going to move through it is DNA. So then let's look at these 3 micro test tubes. Let's say that these are 3 different people's DNA. Oh,
Here's direction for bus change, bus 25. 25 will be here for 31. I'm sorry, bus 31 will be here for 25, and it will be about 30 minutes late. Once again, bus 31 will be here for 25, and that bus will be about 30 minutes late. Thank you.
so these 3 people's DNA, let's look at, when it says here, it's a mixture of DNA molecules of different sizes. So why they're different sizes is because we take each one of those 3 people's DNA and cut them. They're cut with, with the same restriction enzyme. And so our DNA gets cut up into smaller pieces. So what does that mean? If this is person number 1, let's say in their DNA, and here's person number 2 and their DNA, and here's person number 3 and their DNA. Because everybody's DNA is slightly different, we're going to have different cutting sites. So where that restriction enzyme cuts is going to be different for all 3 people. So let's say person number 1 has a cutting site right here, and you end up with three fragments. Person number 2, let's say, only has 1 cutting site, and they end up with 2 longer fragments. And then let's say person number 3 has 4 cutting sites, and so they end up with 5 different fragments. Some of them really small, some of them larger. All right, and so everybody's DNA is going to be cut differently because the order of bases is different, and so therefore the number of cutting sites are different. And so what that's going to let us do is compare DNA using this gel, which I'll talk about tomorrow. All right? So what we have left, you guys, is I just had to, this is the last new technique of DNA technology, and then the rest of in the notes is just things that we use it for. All right. So we use this for paternity testing, for figuring out father and babies.
Just wonder. Yeah. We're going to keep learning stuff after the 8th test. Until the end of the year.
What I